Well, good morning, everyone. This is our EM-X for December, the last of 2025, and we will resume again. I think the second Monday in January is our normal way of doing things. But this is the last one of 2025. I can't say it's been a great year, but we have a lot to look forward to. We have, of course, the World Cup in the United States next year, and the World Cup of Electron Microscopy. International Microscopy Congress 21, IMC 21 in Liverpool, UK. First time it's been in the UK. And also it's a special thing for me because I'm from Liverpool myself, born and bred. And so it'll be great to go back. And I think the papers are now open for submission until the beginning of March. Anyway, without much further ado, I'd like to introduce Hua, who will introduce our biological speaker, and then we'll go over to China, where we have another great speaker, one of the younger generation of new microscopists. So, Hua, over to you, please.
Okay. All right. Thank you, Bob. Good morning to everyone who joined this seminar. It's my great honor this morning to introduce you to Professor David Eisenberg from UCLA. Dave is a professor of chemistry, biochemistry, and biological chemistry at UCLA. He also holds the Howard Hughes Medical Institute investigatorship, and he has been serving as a director of UCLA DOE Institute of Genomics and Proteomics. I have known Dave for a long time. I always admire his research and energy and leadership in structural biology, and he has done many pioneering work in protein biochemistry and biophysics through different types of technique, including X-ray crystallography, bioinformatics, biochemistry, now more lately on cryo-electron microscopy. And his many excellent works has been recognized by many different awards in many professional society, and he currently has been a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the American Philosophical Society, and the Institute of Medicines. And in the last few years, he has spent full effort in trying to understand proteins that involve in Alzheimer and Parkinson diseases. And he particularly used a cryo-EM technique to tackle this problem and lead into a design of small molecule to combat these kind of diseases. So I think we are very excited to have you, David, this morning to share with us your latest exciting development in this technology that I think many of us really need that eventually. All right, David, it's all yours.
Thank you, Hua and Robert, for inviting me to participate in this series. And as Hua says, our lab applies methods of structural biology to discover molecules, small molecules that disassemble, take apart the pathogenic amyloid-type fibers that are associated with Alzheimer's, Parkinson's, and numerous other diseases. So I'm going to lead you through 5 steps to tell us what we do, as outlined in red on this first slide. First, to get us all on the same page, a brief background on the pathogenicity and structure, stability of amyloid fibers, and then show you how we've used cryogenically trapped structures of intermediates to find out how small molecules can take apart these fibers. And then how we use that structure of a cryo-trapped intermediate to identify drug-like small molecules that disaggregate or take apart tau fibers, which are associated with Alzheimer's disease, and also fibers of alpha-synuclein associated with Parkinson's disease. A little bit on the mechanism of how they work, and you will see the surprising, at least to us, mechanism that they work. These small molecules form fibers themselves. We call them mock amyloid fibers. And then these fibers of the small molecules take apart the pathogenic fibers. And then if I go fast enough, I'll also tell you about 7 residue peptids that do much the same thing to disaggregate tau fibers. So to start my little summary of the background, there's some 50 diseases, including Alzheimer's, Parkinson's, ALS, that are each associated with a particular protein in the fibrous amyloid state. You see some such of these fibers in the background here. They are very stable. And at least to this point, we lack any effective drugs to halt progression of these diseases. And the ideal modality is a small molecule, because we can't use antibodies to treat 7 million people in the US with Alzheimer's disease, or 1 million people with Parkinson's disease. We need a small molecule that patients can take at home. In some of these diseases, such as transthyretin amyloidosis, the pathology is clearly a toxic gain of function. The fibers are destroying the function of cells. In others, pathology may be a loss of function, and in many cases it's not known. I was careful to say that these diseases are associated with fibers, as opposed to saying they're caused by fibers, because 400 years ago, the physician and philosopher John Locke, he taught us that, and he believed it's impossible ever to separate association from cause and effect. But I believe in the case of amyloid fibers, the case for causation is pretty strong. And here's one of those arguments. There are hereditary mutant or variant forms of the proteins associated with Alzheimer's disease, which, when overexpressed in mice and rats, produce phenotypes that are very reminiscent of the human diseases. And the same is true for variants of alpha-synuclein and Parkinson's disease. So just expressing the protein in an animal produces disease-like symptoms. So I think causation is very, very likely. Now, most of these fibers are extremely stable, even in SDS or boiling. So the question is, how can you find a small molecule or a peptid that can take them apart? As far as structure goes, all amyloid fibers share what is called a cross-beta scaffold, in which the protein chain, whatever it is folded in a 2-dimensional layer, and then those layers are stacked up, always 4.8 angstroms apart along the fiber direction, because the backbone hydrogen bonds of the amide groups produce that distance. So they're beta sheets that run all the way from the bottom to the top of the fiber. And each stack is called a protofilament. This amyloid cross-beta scaffold is self-templating. That is, once you have a fiber, if there are other monomers present, they add on to the fiber. And that's what is thought to be the reason that there's prion-like spreading of fibers throughout the brain. Once you have fibers in the brain, then they slowly take over the brain by this spreading, the self-templating nature. The individual layers of these fibers are stabilized by zipper-like interdigitation of the side chains of neighboring sheets. I'm going to show you what that looks like in just a moment. And that challenges the ability of potential drugs to take them apart because they're so stable. So here's a simple, this is a electron diffraction structure of a segment of alpha-synuclein. 2 sheets, you can see the side chains are interdigitating, forming what we call a zipper or a steric zipper. As we rotate this fiber around its axis, you'll see more clearly the zipper-like nature. There it is, like the teeth of the zipper holding these two sheets together. So the stability comes from these mated sheets, the hydrogen bonds, which turn out to be highly polarized, and then stacks of side chains on the surface, like tyrosine, phenylalanine, and glutamine. So what's the best target for a drug for Alzheimer's disease? Well, there's a lot of evidence from others that that target is the protein tau, and tau forms fibers. And in work, again, by others, and here's a wonderful paper by the lab of Bruce Miller at UCSF, in which they've, in many patients, measured the brain atrophy over time, and that's by nuclear magnetic resonance, and that's graphed on the vertical axis, and on the horizontal axis is graphed the presence of tau fibers as determined by PET, and you'll see there is a correlation, and even in individual patients they see this correlation. So it looks as though there's good evidence that tau fibers are the target. And another reason is the structure of tau fibers from patient brains, first determined by Fitzpatrick, Shears, and Goddard 2017, but now in numerous labs, including ours, there are many, dozens of these structures of the fibers, and they're always the same structure. It's this double protofilament structure. We're looking down the fiber, and we're seeing an individual layer which has two bent tau sequences, and then they're called paired helical filaments. So there's reason to think every Alzheimer's patient has this structure. If we can prevent it or stop it from growing or get rid of it, maybe we can stop progression of the disease. So both these findings suggest if we can inhibit or disaggregate these paired helical tau fibers, we can halt or maybe even to some extent reverse disease. So how can we do that? Well, postdoctoral fellow Paul Seidler, now on the faculty of University of Southern California, what he did was to use the molecule EGCG, which is abundant in green tea and has been known for decades to take apart amyloid fibers, but it's failed as a drug in many clinical trials, and you can see why. It's just bristling with hydroxyl groups, hydrogen bonding donors, and acceptor, 8 donors, 11 acceptors. It doesn't get into the brain at all, so it fails as a drug, but it does take apart amyloid fibers. And here are tau fibers from the brain of a patient, and in 24 hours, almost all the fibers are gone. Only one did see in against hundreds of cases where there were no fibers. So his idea was to cryo-trap the process of disassembly after just a few hours. So he chose 3 hours of treatment with EGCG and to cryo-trap that structure to see how EGCG was working. And this is what he saw, that the EGCG stacks in columns, essentially in a fiber itself, at the crevice between the two protofilaments of tau. So this is 3 hours. If he lets it go for 24 hours, there are no fibers. So these molecules are stacked in these polar clefts. There's twofold symmetry you can see here. The EGC molecules are spaced at 4.8 angstroms along the fiber axis because there's one EGCG for each tau, bound to each tau. And they're hydrogen bonded in place. But that 4.8 angstrom spacing is greater than their minimum energy separation of the rings of EGCG, which, as Pauling taught us, is more like 3.5 angstroms. Thus, the initial stacking is metastable. And our hypothesis is, based partly on molecular dynamics, that the EGCG initially bound, so the rings are 4.8 angstroms apart, undergoes a transition, and that transition exerts a torque on the fiber, which helps break it. So what good is this in identifying small molecules that can take apart the fiber? That's what we want. So that job was taken on by Kevin Murray, a former MD PhD student in the lab, and now he's a resident in neurology. And what Kevin did was to dock a library of compounds in this, what we might call a pharmacophore, the place where EGCG binds. And his library included 60,000 compounds from a library which is said to be central nervous system compatible. That is, these are molecules that are apolar enough and have characteristics, the Lipinski rule of 5 and other characteristics, which predicts that they can go into the brain. So he docked each of these compounds in this site. He used 2 different docking programs, and in each of them, his positive control is EGCG, and it docks better than 2 standard deviations below the mean. And the compounds he's most interested in are those that bind in this crevice even more tightly than EGCG. And he was able to order 46 of these compounds and then do experiments on them. And here are some of those structures, which are all somewhat similar. And indeed, some of them, like CNS11, when applied to brain tau, broken up. You can see here where it's quantified. EGCG really gets rid of all the fibers in 24 hours. CNS11 does a pretty good job. Others do less well. So our first hit here is CNS11. Now we have dozens of these compounds, which I'll get into later. So Kevin Murray then tried to use CNS11 on alpha-synuclein fibers of Parkinson's disease, and he found that it got rid of some of the fibers, but an analog of it, CNS11G, which you can see on the right, did an even better job. So there's some specificity in these small molecules. The CNS11G in a worm model of alpha-synuclein aggregation, this is C. elegans, which overexpresses a alpha-synuclein labeled with fluorescent compounds that form puncta after a few weeks. And you can see when he adds to the food of the worms, many of the puncta disappear with CNS11, but almost all of them do with CNS11G. So CNS11G is the compound that seems best to work on alpha-synuclein associated with Parkinson's disease. We find that these CNS11G inhibits the fiber seeding, that accounts for the prion-like spread. It protects neuronal cells against toxicity of alpha-synuclein, and in the worm model, it gets rid of those puncta. So how does it work? That's the work of former graduate student Zhou Lu, now a postdoctoral fellow at Northwestern Medical School. So here is CNS11G. See the molecular weight's just below 400. So she added it, CNS11G, to a brain extract of a patient who had a disease, multiple system atrophy, which is another alpha-synuclein disease with symptoms quite similar to Parkinson's disease. It's a Parkinson's type disease. And the next day when she looked, instead of getting rid of the fibers were there, suddenly there were many more fibers. What a puzzle. Here she added the compound to get rid of the fibers, but there were more fibers. Well, what were these fibers? So she set out to determine their structure. I should say that they disappeared again after another couple of days, but she was able to determine the structure. And it turned out, to our amazement, that that fiber had a structure that's composed entirely of the small molecule. There's no protein in it at all. And this is a cross-section of one layer, and there are 23 protofilaments of CNS11G molecules stacked on one another. So it turns out it's a small molecule fiber. Also, I want you to note that it has a right-handed twist. That's important later on. Why does it have the same spacing of 4.8 angstroms per layer as amyloid fibers? The reason is it has an amide group, which you can see here. Here's the amide group, and it forms the same interlayer hydrogen bond that amyloid fibers do. So we call these mock amyloid fibers. So small molecule forms mock amyloid fibers. So then Zhou Lu formed, as I showed you with the tau work before, she formed a cryotrapped structure of CNS11G in alpha-synuclein fiber, an MSA alpha-synuclein fiber. And here is the CNS11G. This is one layer. Now we're looking down a fiber of alpha-synuclein, and it's also a fiber of CNS11G. It's a dual fiber. So it's trapped at 4 hours. If she lets it go 24 hours, then almost all the fibers disappear. Actually, it only takes a little over 12 hours, and both fibers disappear. So they're metastable. So then she pursued this by determining the structures of cryotrapped material at 4, 6, 10, and 15 hours. And in each one, there's the structure of the small molecule, which is faded out here, but it's here. You can see it here. You can see it in each of these. I hope you can see it. CNS11G in each of these. If she waits longer than 15 hours, well, then there are no fibers. And when each of these structures is compared to the structure without any small molecule, you can see that they diverge increasingly from the untreated fiber, as though the fiber's already starting to unfold under the treatment of CNS11G. So through other work, we've learned that the standalone fibers that I showed you that were first seen, those fibers are more stable than the ones that are complexed with the alpha-synuclein protein. So the alpha-synuclein protein fibers are metastable, and they want to relieve that strain by collapsing into the stable structure. So that's what we think is important in taking apart the fibers. Also, there's another important point, and that is the stable structure that I showed you before is a right-handed twist, whereas the metastable structure in the alpha-synuclein complex is left-handed. And so the handedness is part of the lack of stability. All right, so now I'm just going to quickly show you that peptids also exhibit similar behavior. And this is the work largely of Dr. Ke Hao, who is holding an award here, which she received for this work. She found that a 7 residue, all D configuration peptid, that is, as opposed to L configuration, it's a D configuration peptid with this amino acid sequence TOK-IVW, and then a 7th residue, which can be almost any of the coded amino acids, isoleucine, valine, alanine, and so forth, but not proline. Almost any of these disassemble fibers of tau, but not proline doesn't do it in the 7th position, and just the 6th residue position without peptid without the X, that does not disassemble. This work, which I'm not going to go into how she discovered it, it was a meticulous observation and reasoning. It was described last year in Science Advances. So each of these peptids, these 7 residue peptids that disassemble, they themselves form fibers. Whereas the proline one does not, nor does the 6 residue peptid. So there's a correlation. The peptids which form the most fibers are also the peptids that are best at disassembling tau fibers. And these peptid, all of these peptid fibers are right-handed. They have a right-handed twist, whereas tau fibers and alpha-synuclein fibers are left-handed twisting. Here's a time course on electron microscopy of adding this TLK-IVWI, this is the isoleucine variant, to tau fibers that come from the autopsied brain of an Alzheimer's patient. You can see these thick fibers, which are the brain tau fibers. And she adds now the 7 residue peptid, and after about 3 hours, now other sorts of fibers appear, much thinner fibers, along with the thick ones. And by 6 hours, the more of the thin fibers, fewer of the thick fibers, and by 24 hours and 48 hours, almost all fibers have disappeared. So when the 7 residue peptid is added to the pathogenic fibers, a new fiber appears, which is presumably the peptid fiber, and then it takes apart the pathogenic fiber and itself as well. She determined the structure of the complex of pathogenic tau fibers complexed with a peptid, and the peptid forms a steric zipper on the surface. It's a fiber. We're looking down the fiber. It's a steric zipper structure on the surface of the tau fiber. Again, a dual fiber, but this is a strained peptid fiber because it's left-handed, whereas we know when it forms on its own, in the absence of tau, it's right-handed, but in this case, it's left-handed. So now I'm going to show you in real time by the method of atomic force microscopy how the peptid, this 7 residue peptid, takes apart a tau fiber. So in atomic force microscopy, there is a cantilever with a very narrow, almost atomic scale tip, and the cantilever taps all over the surface of the sample. It has a mirror surface, and a laser focus on it bounces off the tip, and when changes in height are recorded by a photodiode in the wall in the next room. And so small motions are recorded, and the higher the displacement of the cantilever is shown by a brighter color here. So that bright color means a little hill in that surface. And in this sequence, there is a fiber of tau here that seems to be a bent fiber of tau, and it's now been treated after 6 hours. It's being treated with the peptide TLK-IVWI that I showed you earlier, and we're going to follow that over 12 minutes, except it's speeded up. We don't have 12 minutes here. And here's the start. The peptide is added, and you see the fiber breaks up into smaller pieces, and then those pieces break up into even smaller pieces. And by 737 seconds, that's 12 minutes, almost all of the fiber has been broken up to a size which cannot be distinguished by atomic force microscopy. So here's what we think is going on in cartoon form. Here's the peptide or small molecule is added to the pathogenic fiber. It's nucleated on the surface. The concentration is high, so it binds to the tau fiber and forms a nucleus. Because it's ordered, the entropy goes down, so the free energy goes up. But once it's nucleated, now other fiber molecules or small molecules add easily to the fiber, and they bind, so the energy goes down. But as it continues to grow, now the fiber is metastable, so it exerts a strain partly because in this case it's the wrong-handedness, but also for some other reasons. And eventually that strain builds up so that it becomes equal to the energy of breaking the fiber, which we estimate to be about 25 kilocalories per mole. So here's a point of where it's going to break because the fiber has grown enough to have a great strain. Now it snaps, and both fibers snap into pieces, and these are the products which don't reassemble because they're still bound to fibers of the peptide or the small molecule. So just to summarize, these structural methods have identified both small molecules and short peptides, 7 residues, that disassemble these very highly stable pathological amyloid fibers associated with Alzheimer's and Parkinson's diseases. Both the disassembling small molecules and short peptides themselves form amyloid-like fibers. We call them mock amyloid. They're nucleated by monomer binding to the pathological fiber. The complexes are in metastable states, creating a strain, and our hypothesis is that the drive to relieve the strain in the complex is the source of energy for disassemble of the fibers. And finally, I just show you the faces of the scientists who've done this research. Paul Seidler, now at USC Medical School. David Boyer, expert in cryo-EM structures. Zhou Lu, who discovered these mock amyloid fibers. Ke Hao, who did entirely the work on the peptides. Kevin Murray, who discovered CNS11 and CNS11G. Michael Sawaya, our computational guru, and Lisa Luder, who's our AFM specialist. And thanks to our funders, and especially to the families who donate brains, and to the pathologists. And I thank you for listening.
Wow, David, thank you very much for these fascinating stories, how elegant experiment you did, and also with your colleagues, and all these results provide chemical mechanisms of how this molecule works on these fibrils. So I think there are some questions from a Q&A. And then the first questions is there any effect on the rise and twist of the fibers during the interaction with the CNS11?
Is there any effect could you repeat that?
The rise and twist of the
Yes,
fibers.
yes. So the twist in the complex structures is always the twist of the pathological fiber. Because those of you who've used Reliant or other helical averaging know that the structure you see reinforces features of that obey the same helical parameters. And so we see the small molecules and the peptids in these complexes, and they are obeying the same twist of the pathological fiber, namely left-handed. Yet we know when we look at the standalone fibers that they are right-handed. So, and that's their preferred low energy structure. So it's the strain of the fiber that's forced into its left-handed twist, which is the feature which causes the metastability.
Okay, good. Thank you. And another question related to CNS11G. Does it form much stable filament structure than the alpha-synuclein ones? Then how does it disappear after a few days?
Well, that's a very good question, and I don't have a complete answer for you, but I can say that the small molecule fibers form only when seeded in a brain extract. That is, if we take CNS11G and put it in various buffers, it doesn't form fibers. But if it's put into a brain extract of Alzheimer's or MSA, now it forms fibers. So it has to be seeded. And now, then I suppose what happens is that it now complexes with the pathological fibers that are present in that brain extract, forms the metastable complex, and then both fibers snap, and that's why it disappears. That's my hypothesis.
Great. And another question is, when the protein fiber disassemble, what happened to the monomer that come out the fiber? Do they get degraded or they come back together later on?
Another excellent question. So what we know about the products are that they don't reassemble into fibers, and they're no longer seeds, and they're too small to see in negative staining electron microscopy, but they're still quite large. When we do mass spectroscopy, the median size is something like 50 kilodaltons. So these are quite large pieces of a pathogenic fiber now coated with the small molecule or the peptide. So they're not monomers.
Okay. And another question is any information about the structure of these small molecule that would affect their access across the blood-brain barrier?
Just that the small molecules are apolar to begin with, but now we've started experiments in mice, and we find that the concentration in the mouse brain is small but significant. It's in the nanomolar range. And in the one preliminary experiment, it seemed to have effectiveness in combating pathology. So it does get into the brain.
Okay. Another question is the physical mechanism of these aggregations purely physical force or is caused by the decrease of the conformational entropy or both?
Is it a physical force or
decrease in the
increase
conformational entropy?
I would, it's another good question, and I have no data to answer. My guess is it's a combination of both.
Yeah. Okay. And then another question is, what is the resolution of the map you talk about?
Yes.
Yeah.
So of the maps that I showed today, the very best resolution was 2.8 angstroms. For the peptide complexes, they're more like 3.3 angstroms resolution.
Okay. All right. Good. There are some remaining questions from this questionnaire, but let me jump to another one from Tom Polar. That he thought that the assembly intermediate contribute to the pathologies. So how are the partially disassembled fibril a problem?
You'll have to repeat that. Wow.
Yeah, okay. So the assembly intermediate contribute to the pathology, right? And how about these partially disassembled fiber? Would that be a problem? Is that also cause pathological
Oh,
problem?
partially disassembled fibers.
Right,
Yeah.
right.
Well, what I can say is that when we treat the fibers with small molecules and then test them in the biosensor cells of diamond, that they largely halt seeding. So and the toxicity goes way down. So they seem to inactivate the toxic features of the fibers. Not completely, but largely.
Okay, good. Thank you. There's still many questions. Let me just raise one last question, and some of the questions can be answered after the second speaker. The last question that raised by Ed Egerman is, how do you determine the handiness of those fibrils or the amyloid?
And by AFM, atomic force microscopy.
On the handiness of the fibril?
Yeah. Yeah.
Okay.
Yeah, we got nice AFM images.
Okay. In the same sample, you prepare for the cryo-EM.
Right. And of course, once you get to atomic resolution and can see the carbonyl groups, then you know the handedness as well, if you have sufficient resolution.
Okay, and your 2 or 3 angstrom resolution, you are confident about this handiness?
I would say at that resolution, yes.
Okay.
But on the whole, we've used AFM to determine handedness.
I see. That's interesting. Okay, so David, thank you very much for your excellent presentations and all the outstanding questions being raised by the audience. And there's only a series of questions which hopefully David can stick around after the second speakers to continue the informal discussion. So I turn back the podium to Bob to introduce our second speaker. Bob.
I have to stop sharing.
Yeah, please.
It's okay. I wanted to ask David something, if I may, which is, when do I start taking this stuff? Sounds brilliant to me. So,
well, as you know, there are many, many steps to drug development, and we're on our way with mouse studies, but there's still many steps.
Okay, if you need a volunteer,
maybe start drinking the green teas to start with. That may help.
maybe that's it. Yeah. And talking about green tea, we should move on then. Thank you, David. It's brilliant stuff. To our next speaker, who's calling in from China. So it's after midnight now. And so I want to thank Wu for agreeing to do this for us. And also thank Jane and Prasanna for wonderful organization of this EM-X this time. And to thank Mr. Yi Che, who has now graduated and has been helping us do this for 3 or 4 years now. And so congratulations to Yi for moving on, passing his PhD, and going to University of Chicago for a postdoc. So I'd like to just say a few words about Wu. He is graduated from Tsinghua University and went to Lehigh for his PhD with Chris Kiley, and then on to Oak Ridge, and had a prestigious Wigner fellowship there, working with Pantelides and Steve Pennycook, and Juan Carlos Idrobo as well. So then he returned to China at the University of Chinese Academy of Sciences, and as I said earlier, one of the new generation of brilliant electron microscopists there. There were several really great talks at our recent IM Nano, both in Japan and Sweden, and he gave one of them. So I'm very pleased to introduce Wu Zhou, who will give us the next talk. Please, Wu.
Okay, thank you, Bob. You hear me okay, right?
Yes.
Yeah, okay. Well, it's a great honor to be invited to the EM-X symposium. And as Bob introduced, I just came back to China 10 years ago, exactly 10 years ago. And we have been working on vibrational use since the installation of our microscope. So today I'm going to introduce to you some of the recent works. We use vibrational use to try to do isotope mapping at the atomic scale and try to probe electron-phonon coupling in a superconducting oxide interface. So just a little bit introduction of, let me see if I can move. Vibrational use, and this starts about 11 years ago when Andre Krivne introduced a microscope that could achieve very high energy resolution, about 14 at that time, and right now I think the world record is about 3 milli-electron volt. So in the spectrum, they can start to see a peak associated with lattice vibration. And over the past 11 years, we see a lot of exciting progress in this field, owing to the high energy resolution of the electron microscope and the spectroscopy method. From the very first vibrational EELS back in 2014. And nowadays, this year, we see very exciting results from the SuperStem group detecting magnons in materials, and also from the Peking University group, Penghao's group, doing in situ thermal resistance measurement at the interface with a device inside the electron microscope. So based on those exciting progress, we just want to do two things. One, the first world I'm going to introduce to you relies on the high spatial resolution combined with high energy resolution, and also trying to do that with single atom sensitivity. We'll try to see if we can do the isotope mapping atom by atom, and those results are done by my students listed here. To start with, we just very brief introduce how we can do that. Measuring the vibrational signals in the electron microscope, we can collect the signal through two different mechanisms. One through the dipole scattering. This is mostly done on polar materials where the electrons will excite the phonon, and we collect it with very small angle, and those signals would be very strong, but their interaction distance is very large, so it's lack of spatial resolution. And the other way is that we use the electrons to hit the atoms directly, and this will excite the vibration of the material. And in this way, the incident electron will scatter to a high angle, and those high angle scattering will give us very high spatial resolution. So this is the mode that we are going to primarily use in the following studies. And the results actually based on one of the very important work 5 years ago published by the SuperStem group. What they demonstrate here is that using vibrational EELS or phonon EELS, they can actually sense the difference in the local vibrational signals introduced by a defect. In this case, it's a silicon defect embedded in the monolayer graphene lattice. So they compare the spectrum acquired from the pristine lattice and acquired from this point defect. So they can see the very subtle difference. And this very subtle difference is, can be reproduced by the calculated phonon density of state projected onto different atoms. So this is the very first demonstration of vibrational EELS could achieve single atom sensitivity. But here, from their calculation, they actually see that apart from this silicon atom, which show very different signal from the graphene lattice, the carbon lattice around the defects also show energy shift. But in the experimental data, it's very difficult to see. So in order to see this, we actually first need to go back to the very simple model, a harmonic oscillator. We can use this model to describe the vibrational frequency. So the frequency will depend on the spring constant, and at the atomic scale, this is the chemical bonding strength, and also depends on the weight of the ball, which is the atomic mass here. So in order to decouple these two contributions, we need to have a very stable microscope and very high signal to noise ratio in the spectroscopy data. So we can look at the spectroscopy data from each individual atoms around this point defect. So here I just show you that over the past couple of years, we spent a lot of efforts trying to improve the stability of our microscope. And this is a spectroscopy mapping taking over 20-30 minutes. And you can see that from a single frame acquisition, we can still have very good result of the carbon lattice with minimum distortion. So this type of stability will allow us to extract the spectroscopy from each individual carbon atoms and to compare their frequency and see how it's affected by the chemical bonding change when we're looking at the defect sites. So this here is the spectroscopy from graphene showing that we have two peaks here corresponding to the acoustic phonon and optical phonon modes of the graphene lattice. So in this study, we actually show that when we have a silicon atom in the graphene lattice, and if you move away from this silicon atom, and just by looking at the carbon atom, and because the chemical bonding will change from, they are all as three-fold bonding, but this nearest neighbor we label in red is bonded with carbon and silicon, and the green atom we label here only bonded with free carbon. So you can actually see that we can have the sensitivity to see this very small 3 milli-electron volt energy shift in the acoustic phonon mode, and it gradually blue shift towards the pristine graphene lattice. So if we compare across two types of defects, one is the three-fold silicon defect, another is four-fold planar structure, and in the three-fold case, it's a barcode structure. And also we're looking at how the carbon signals change when you go away from this impurity atom or defects. We can see that they're actually quite different. One is blue shift, and the other is a gradual red shift. I'm not going into the details here, but basically this is affected by the chemical bonding difference. So this is three-dimensional bonding, and this is a quasi two-dimensional bonding. And we see that the chemical bonding actually affects the local vibrational modes, and we have the sensitivity to pick it up. And the other factor, as I mentioned, affecting the vibrational frequency is the mass. So in this case, we change the impurity atom to a nitrogen atom, and we can pick up the spectrum from the nitrogen atom and from the neighboring carbon atom. We can see in this case, it's the optical phonon mode will shift. And if you just measure the frequency, it matches with what you can predict with a simple harmonic oscillator model, square root of the mass ratio. So that means that we actually see both, we can decouple both. So if we take this one step further, the question is, can we measure the smallest atomic mass difference? So that would correspond to the isotopes, carbon-12 and carbon-13 difference just by one atomic mass. So what we see here is that if you grow your graphene with pure carbon-12 methane or pure carbon-13 methane, the optical phonon modes, you can actually see that they're located at different energy. And this is consistently also being picked up by Raman spectroscopy, for example, and also demonstrated by Kato Surinaga's paper published in 2022. So the ratio could be just, as I said, described by the atomic mass ratio, a square root of the atomic mass ratio, described by the harmonic oscillator model. So this is done at the macroscopic scale, or in Kato's paper, it's done at the sub-nanometer scale. In electron microscope, so that's already the best, almost direct world record. That's a sub-nanometer scale. They can can map out the distribution of carbon-13, carbon-12 in the carbon-13 matrix. So the question we raised is that, is it actually possible to identify the isotopes atom by atom? So that would be the ultimate goal for structural analysis. So let's first see whether it's possible. So to do this, we understand that the frequency, the vibrational frequency, is mainly determined by the atoms you are looking at. But from the previous case, we also see that it's also affected by the neighboring, especially the first nearest neighbor atoms bound directly to the target atom. So here we label, calculate using DFT to calculate different type of structures. That the white atom is carbon-12, the black one is carbon-13. So we have carbon-12 bonded with different number of carbon-13 atoms, and here's the carbon-13 bonded with different numbers of carbon-12. So what we see is, if you're looking at the optical phonon mode, and just looking at the p position, we see that carbon-12 center atom always have a higher vibrational frequency. When you are looking at carbon-13 atom, no matter how it bonds with the neighboring atom, you always have a lower frequency. But the difference between these two is small. So that means that if we want to do atom isotope analysis, we need to have plus minus 1 milli-electron volt accuracy in identifying the p position. And this is very difficult to achieve if you start with a spectroscopy data like this. And this is the typical signal to noise ratio we can achieve when we do the mapping. So in order to get to high accuracy from locating the p position from this type of level of signal to noise, one of my students, Zhou, he's right now a PhD student at Cornell. What he does is to develop a neural network to denoise the experimental data. So start from the noisy data here. After denoising, we can have a reasonably good signal to noise ratio in the spectrum. And from there, we can accurately identify the optical phonon p position. And here's just a test that after applying the denoising process, we can go from a broad distribution where we mix the carbon-12 and carbon-13 spectrums and to a very high accuracy analysis. So with this process, we can looking at isotope mixed sample. So the first one we look at as carbon-12 and carbon-13 interface, just by switching the methane source precursor when during the growth. And you can first identify the macroscopic interface using Raman mapping and then put it into the electron microscope. So from the image along ADF image, we would not expect that we can tell the difference from carbon-12 and carbon-13 because this mode, image mode, is only sensitive to atomic number. But if we map out the optical phonon p position in this area, we can actually see that the top part, it has a higher frequency correspond to carbon-12 and the bottom part corresponding to a heavier atom, carbon-13. And we see a switch just across this carbon-carbon bond. So we have the spatial resolution better than 0.2 nanometer here. And we can go one step further to look at some area where we start to have isotope mixing. And we can identify that here we have a carbon-12 in the carbon-13 part. And you can see that from the raw data and also from the denoise data where we identify the p position and assign to different configurations based on the DFT calculated p position. So in this way, we actually demonstrate that we can identify individual carbon atoms in the carbon graphene lattice with its isotope identity. And we go one step further to test whether this whole process is the accuracy of the process. And we use isotope mix sample just by mixing, during the growth, we mixed 20% carbon-13 methane with 80% carbon-12 methane. And Raman measurement tell us that in this area, we should roughly have 20% carbon-13 in the micro scale. And in the atomic scale, across different region data sets, we actually get to very consistent results. So that's a strong indication that we actually achieve atom by atom isotope mapping, at least in this monolayer graphene system. So this is the first example I'm going to share with you that we can try to push the limit of this vibrational EELS method and try to do single atom isotope mapping. And the second example I'm going to show you is going to use, again, the high spatial resolution, high energy resolution, but combined with momentum selection during the experiment to understand electron-phonon coupling in a superconducting oxide interface. So the system is very different from the previous example. In this case, it's an insulating oxide, lanthanum alumini growing on top of strontium titanate. And this sample is grown at high temperatures of the LAO layer that's crystalline. And both materials, they are insulating, but when you grow them to form this interface, they will form a 2D electron gas due to the electron transfer from the lanthanum oxide layer to the STO region. And it will change from, upon tuning the charge doping, you can tune this from an insulating oxide to a superconductor, although the TC is extremely low, about 200 to 400 millikelvin. In this system, the superconductivity can be switched on and off just by gating to tune the charge carrier density. But our collaborators find out that you can also tune the carrier concentration by changing the lanthanum aluminum ratio at the LAO layer. So we'll get back to that. So we want to see how in this particular system, the 2D electron gas would induce new interfacial phonon modes, and how this will correlate with inversion symmetry breaking, and also more importantly, the emergence of superconductivity in this insulating interface. So if we look back to the literature, last year there are two very important papers, one published by Professor Xiaoqing Pan's group at UC Irvine, and the other almost at the same time published by Professor Peng Gao at Peking University. Both papers are dealing with superconductor interface, monolayer iron selenide growing again on STO. But in this system, the STO layer will reconstruct at the interface, and this will correlate it to enhanced TC in the iron selenide layer. So using vibrational EELS, and also they probe it with momentum selection at the outer plane, vertical to the perpendicular to the interface, or parallel to the interface with momentum parallel to the interface, they can see localized phonon modes that's just localized at the interface, and this is also reproduced by the calculation. So these two studies, they show that localized phonon modes would emerge at this reconstructed interface of STO and will influence the superconductivity transition in the iron selenide layer. So we basically applying the same method to looking at our system, but this case we try to understand how the two-dimensional electron gas will affect the local vibrational modes and correlated to the superconductivity. So we have, as I said, we have 4 samples. In this case, because we can't do electrostatic gating inside the electron microscope, so we use the LAO layer to control the charge doping. And in this case, it's specifically by the lanthanum aluminum ratio. You can change the charge doping from 0.8 to all the way to 2.5 times 10 to the power of 13 electrons per centimeter square meter. And this will tune the sample from non-superconducting all the way to almost optimal doping level with a TC of about 300 millikelvin. So we'll use this sample, these 4 samples, to look at several variables. One is the charge carrier density, and the other is the electronic reconstruction at the 2D-dimensional electron gas, and then correlated with the structural symmetry breaking and the interfacial phonon modes. All quickly done on the same microscope during the same experiment. So first we look at the electronic reconstruction introduced by the 2D electron gas. In this case, because lanthanum transfer to electron to the STO layer, the titanium oxide layer, will partially reduce the titanium 4 class. We can see this actually from the titanium L edge. And you can see that going from the bulk all the way to the interface, there is a very small shift, about 30 millielectron volt on the EG energy level. So if you map out the distance between the T2G and the EG, you can actually see that with the increase of charge carrier density, we see a progressively increase of the 2D electron gas layer thickness. And we can also see that from the oxygen K edge, where you see this shoulder peak will gradually increase in energy and merge all the way to the main peak. And this is an indication of the structural distortion, more specifically TiO6 autohedral distortion. So this is an indication that we have structural distortion disparallel due to the two-dimensional electron gas. So this is from the electronic structural reconstruction. And we can also see from the structural analysis, atomic structural analysis itself. So in the low doping structure, we have antiferro distortion combined with a polar distortion. So you can see the oxygen position from up, down, up oscillation. And you can also see that this distortion from the superlattice spots. But if you map it out, the lattice parameters, and you can also see that with the charge doping increase, the structural distortion region also increase. And that's very consistent with the thickness increase of the 2D electron gas layer. And at the optimal doping, you see actually the structure develop into a polar structure with the structural model we shown here. So we see that the electronic structure correlates with the symmetry breaking at the interface. And the last point, missing puzzle, is to look at the phonon modes. And I'm not getting into the details here, but basically we can move the electron, shifted the diffraction pattern to make the electrons getting into our EELS aperture with momentum either parallel to the interface or perpendicular to the interface. And we can also shift it 45 degrees to collect both in-plane and out-of-plane vibrational modes. So we're just looking at the results. So the first scenario is when we use a QXZ geometry, we collect both in-plane and out-of-plane vibrational modes. You can actually see that with increase of the carrier density, we see this change of the spectroscopy data. We don't need to worry about the details, but you actually see that the changes progressively increase into the STO layer. And this is a strong indication that the phonon modes are affected by the charge carrier. And if we plot it by line cut, we can actually see the peak positions more clearly. You can see how it shifts from the bulk in to the towards the interface, which is the green curve here. And again, if we assign different modes, and this is what we label P2 and P4 in the literature, they are actually polar modes introduced by the polar distortion related to the optical phonon. And these two modes, if you're looking at them, the energy shift and the intensity increase is more pronounced in higher charge doping and more pronounced when you go towards the interface compared with the bulk. So that's an indication that the polar modes are very sensitive to the confined charge at the two-dimensional electron gas at the interface. So finally, if we looking at the new modes, vibrational modes, emerge at the interface in addition to those intrinsic STO vibrational modes, we need to use momentum selection to just looking at the modes parallel or perpendicular to the interface. So in this case, we're just looking at the highest doping sample, and we look at the vibrational signals atomic layer by atomic layer. So here what you will see is that we see a few additional new modes that's localized at the interface, and it fades away when you go just like 4 unit cells away from the interface. And this corresponds to the outer plane vibration and in-plane vibration when we use different collection geometry. And the interesting thing is the outer plane vibrational modes, you can see that in both the strontium oxide layer, the titanium oxide layer, but the in-plane modes, you only see that in the titanium oxide layer. So it's highly localized. And I should mention that this very short distance is correlated, consistent with what people found the superconducting layer thickness in this system. So we believe this localized phonon modes, they are actually correlated with the superconductivity. And well, I'm not getting into the details here. Basically, what we are seeing here is that we plot the outer plane vibrational modes and in-plane vibrational modes as a function of the charge carrier density across these 4 samples, and we're just looking at the end result here. So what we see here is that the outer plane vibrational modes is the most sensitive to the charge carrier doping. It changes energy by 6 milli-electron-volt across these 4 samples. And those are the experimental observations, and they are actually reproduced by our DFT calculations, which sends these 2 additional in-plane vibrational modes and the additional of these outer plane vibrational modes. And you can see that those modes, they just fade away when you go in the theory. It's just like 2 unit cells away from the interface. And in our experiment, it's about 4 unit cells. And the theory also shows that if we're looking at the vibrational modes for this outer plane localized vibrational modes at about 95 milli-electron-volt, it's mostly contributed by this apical oxygen in those octahedrals. And if the in-plane vibrational modes, it's actually mostly contributed by the equatorial oxygen in the TiO layers. And also we see that both modes are very sensitive to charge doping, and you increase the charge concentration in the calculation, you can actually see how it shifts in energy, and very consistent with the experimental observations. And the final part is that we calculate to verify that actually these 2 modes actually have very strong electron-phonon coupling. We calculate the phonon line width, and you can see that this outer plane vibrational modes and the in-plane vibrational modes, they all have larger phonon line width or stronger electron-phonon coupling. And with the strongest one is coming from this outer plane vibration that correspond to the contributed by the apical oxygen. So here I just give you a brief summary. So we see that in this study for the LAO-STO interface, we use momentum selective vibrational mode used, and we see that the appearance of this localized high frequency outer plane vibrational modes, they are having very strong electron-phonon coupling, and they correlate very well with the inversion symmetry breaking at the interface, and also the electronic structural reconstruction at the 2D electron gas layer. And they also correlated with the onset of the superconductivity at this optimal doping samples. In the previous sample, I show you that we can push the limit of vibrational EELS technique to do atom by atom isotope mapping. So with that, I want to thank my students and postdocs who actually did the work. These few phases contribute, I show you with individual works, and also to our collaborators, so Pantelides group at Vanderbilt doing the theory, and the samples for the LAO-STO provided by people from Barcelona and two groups in Barcelona and also the funding agency. And if you allow, I just want to put one advertisement that we have postdoc positions open. And if you want to work on these two new microscopes, you're welcome to contact me. Here's the barcode to our group website. Thank you very much.
Wow. Thank you very much, Wu. That's really fantastic stuff. And thank you for sharing so much of your new data, which is now under review. And this is really exciting. I have a couple of questions myself, which is how do you obtain such great energy loss and high resolution? Is it you just put it in the microscope and just take the data, or you just have to, what do you have to do special to get these sort of resolutions?
Okay, well, the energy resolution itself, it's the credit goes to Andre Griffney, the neon guys. Their microscope can achieve almost 6 milli electron volt at the best on the microscopes that's already installed. And there are more than 10 such microscopes around the world. And in this particular case, I think what's important is the stability of the microscope. In our lab, we try to we spend a lot of effort try to stabilize the temperature and reduce the vibration of the ground. So this will contribute to the enhanced stability of the microscope that will allow us to take the data with high signal to noise.
Maybe Kate, if you have any comment on that, because you're hoping to set up something equivalent here.
Yeah, we've also been at Stanford thinking a lot about the room design and how to get the best stability to make sure this microscope works at its absolute peak. I had a question also on these like isotope mapping. When you have these blue and red atoms, you were able to do that isotope mapping at the atomic scale?
Yes, let me go back. It's actually, yes, the image you see here is actually the simultaneously acquire ADF,
Yeah.
and we extract the spectrum from individual atom and analyze the p position and assign to each isotope. And then you can see actually it's not that homogeneous across different data sets, but the average is very consistent with Raman and the nominal growth condition.
And how did you, so in your calculations, some of them were like a 1 MeV difference.
Oh, yeah.
Right? And
That's,
as you said, kind of neon microscopes, like maximum 5 MeV, I suppose.
Yeah, that's a very good question. So here, the error bar, it's actually coming from calculation. So this is a very large supercell calculation. So they can even have the same configuration. I didn't mention that actually the second nearest neighbor also contribute a little bit. So that's the source of the error bar here in the calculation. So that means even you have the same configuration, the p position could deviate a little bit. And the second question is related to the accuracy, how reliable you can measure this p position. And you can see that the energy resolution in this case is still about 10 milli electron volt. But the accuracy, you can identify the p position, it's related to the signal to noise ratio.
Yeah.
Yeah.
In fact, there's a question from the audience here just about that. At these scales, how do you manage noise and extract reliable vibrational frequencies from these data?
Yes. So that's I think that's the work, the core of this work that my student developed this neural network to denoise. And we spent a lot of time just to test how reliable it is. And here I only show you one case. So we just mixed the data from carbon 12 and carbon 13 altogether and see if after denoise we can still accurately assign, because we know the ground truth of the data. We can see that with denoise, we can basically 100%, in this particular testing, we can 100% assign carbon-12 and carbon-13. And in some other more challenging tests, the accuracy is consistently above 90%.
So from a materials point of view, there are two things which come to my mind immediately,
Yes.
which is the nitrogen in diamond as opposed to nitrogen in graphite. Nitrogen in diamond is a really important system, but I suppose the three-dimensionality of a diamond specimen is going to really hurt the data there. And the second question is, how about hydrogen and deuterium? I mean, so there's a lot of materials and biological materials which can be addressed with hydrogen deuterium differences.
Yes, very good question, Bob. The first one, as you said, nitrogen center, color center is very important. But in that case, the first challenge is to identify this during microscope experiment, that you need to be able to see that from the three-dimensional structure. So I think there is a recent paper published in I can't remember, in the Innovation, probably that's the journal name, using tachography to resolve the color center structure. But then the second challenge is to probe the vibrational signal signature from there. So it's challenging, but I think it's a very important, interesting system that was trying. The second comment, hydrogen and deuterium. Yes, that would be actually, in a way, it would be easier because the frequency will be much further apart.
Yep.
But it's more challenging in the sense that it would more easily damaged by electron beam.
Right.
But there is actually a very nice paper from Kato Srinaga's group. They map out, they use deuterium to label some of the polymers, so they can see using the hydrogen signal as a probe to map out the different type of monomers in a polymer.
Wow, thank you. So, Kate, did you have something more?
Well, I was just going to say, as Bob mentioned, maybe we'll be connecting with you offline to chat more about how you get these amazing results from your neon microscope and the kind of way that you integrate it into the shared facilities.
Okay, no problem. Yeah. Also, I can send you a paper about our room design, but it's written in Chinese and published in the Chinese Journal of Electron Microscopy. I'm sure you can use, for example, any AI to translate it.
Well, we have enough Chinese graduate students who can help.
Yes.
I'm sure Wa could help as well. So, but anyway, this is fabulous. Maybe, Kate, we should go over to Beijing and just check out what they've done there.
That'd be really cool.
You are welcome anytime.
Right. So I'm sure Juan Carlos is also trying to set up something equivalent as well. So what do you have any comments and engage David back again?
Yeah, there's a few more quick questions for David. Let me just read it out.
Maybe just one more, Wa, because we're getting close to time. Please.
Okay. So David, there's a questions about how you prepare the samples. Do you use a feed milling procedures to prepare your fibrils, or you just put the fibril on the EM grid?
The fibrils from brain are partially purified during the extraction procedure, and then that crude mixture is applied to the grid. So there's lots of material there besides fibers.
I would have thought also that there's a possibility here for using some of Wu's techniques to look at some of these things as well. I mean, those spacings and the differences from what you expect, they must give rise to vibrational effects, which I don't know whether we can capture them quickly enough, though, before we destroy them with the electron beam.
Yes, and we need the stable microscopes that he has.
Right.
Well, in the sense, the vibrational EELS can pick up very similar information as infrared spectroscopy, but with nanometer scale spatial resolution.
Yeah, that's rather different. So, which is why we're doing electron microscopy.
Right. And then, David, have you checked on the toxicity of your disassembled products with the short peptides?
Yes, we have. And the toxicity is eliminated. So whatever comes out of the brain, if it's fibers or oligomers or both, the peptids and the small molecules negate that toxicity.
Mm-hmm. Okay. And there's a questions about the D-peptide with the per molecules. The question is, these D-peptide forming fibrils themselves, are they expect to perform in vivo, you think? Because all your experiments are in vitro.
They are. So delivery, of course, is a challenge. So Dr. Zhou has engineered complexes of the D-peptides with magnetic nanoparticles, which go into the brain. So those would be the next thing to test in a mouse experiment.
Yeah. So that's kind of exciting because your work basically paved the basis to move ahead for the next steps, I guess.
Right.
So to speak. Yeah. Okay. That's fantastic.
Okay, well, I think maybe we should wrap up the formal session now, and we can carry on.
Yeah. Okay. That sounds good.
David can reply to some of the Q&A online as Zoom allows.
Right.
You have any final comments, Wa?
No, I think both talks are outstanding, and I'm particularly thanking David for this work. I think this is really fascinating using the cryo-EM technology in solving some really pathological problems that I think the aging population in the world are facing. So I think it's a very timely application of cryo-EM. And I thank David for taking a lead, and particularly the fascinating story that forming one fibril and with the small molecule and with the peptide, we change its conformation. So I think with all this cryo-EM method, able to review that is extremely fascinating to me. Thank you very much, David.
Thank you, Wa.
And Wa means hurry along so that Wa and I are saved also in due course before it's too late.
Right. So we need to somehow convince the government to provide more funding so to accelerate this process. David, where do you collect all your data on the cryo-EM?
Wherever we can. So
Whenever you can.
yeah,
Okay.
your Slack has been very important to us at UCLA, Northwest, and New York, once at Geneva, once at Caltech.
Okay, real good. You are always welcome, because we want to accelerate your discovery
Well,
process.
that's wonderful. And
Yeah.
Stanford's been very, very helpful.
Okay, thank you.
All right, so let's close the formal session. And before we depart,